Had the opportunity to present at today's webinar and would like to acknowledge the tremendous forum for knowledge sharing in medicine development that the Certara webinars provide. It's important for us at the outset to first state a few disclosures and acknowledgements. We'll be presenting data that is derived from publicly available sources. Any representation or interpretation is our own. It's also key that we acknowledge many who have contributed to the programs, including the clinical trials investigators and participants, and many collaborators and many colleagues over many years and across many organisations. Now, you know a little bit about us, now a little bit about you. Today we have on the call actually more than 200 registrants from 18 countries, with representation from all major pharma, biotech, regulators and clinicians, spanning early career to seasoned scientists and leaders. It really is a diverse brains trust, but connected with a common interest of accelerating the development of medicines in pediatrics. To such an audience, it would really be disingenuous for us to present ourselves as special experts. Instead, it's our objective today as drug development practitioners to share some of our perspectives and experiences in pediatric development, to highlight both opportunities and challenges, and to take today's opportunity to crowdsource some of your insights. We hope that through Certara webinars such as this, we can all keep ourselves motivated to encourage innovation in regulatory science and development to accelerate the availability and optimal use of pediatric medicines. Today's seminar will start with a background provided by Patrick. I'll then describe the case of development and approval of Tamiflu in infants under the age of 1 year, which provides references for further reading around translational and quantitative pharmacology strategies in pediatric development. Patrick will then touch on some evolving approaches relating to new molecular entities for respiratory syncytial virus, where fast-tracking to pediatrics is highly desired, and discuss more general challenges and opportunities to accelerate development of medicines for children and infants. And finally, we'll have an opportunity to hear from you and receive any questions or important insights or perspectives that you wish to share. I'll now hand over to Patrick.
Great. Thank you, Craig. So before we get started, we'd like to set the stage a little with some general guiding principles that I think most will agree are important considerations in developing drugs for children. First, everyone deserves medicines that meet their own individual needs, which includes drugs that are proven to be safe and effective, which have been optimized for dose and dose regimen, and which also meet high-quality pharmaceutical standards. And we know historically, 80% of medicines which are used in children were designed for adults and had little to no data available to guide prescribers on proper use in kids. Secondly, which will be very well familiar to this audience, we need to consider that pediatric patients are not simply small adults, and that there are significant differences when it comes to how diseases both manifest and progress in this population. And as we'll touch on a bit more coming up, there are also significant differences and oftentimes unknowns in terms of pharmacokinetics and pharmacodynamics. And all of this translates into a very different risk-benefit proposition for studying and initiating clinical development in children. Finally, while many impressive advances in this area have been made from the scientific perspective, from the regulatory science and operational sides, it's clear that we need new and better tools. These tools include better fit-for-purpose formulation approaches for children, new approaches to optimize study designs, including things like adaptive designs and Bayesian approaches, which take advantage of data that may exist elsewhere in the literature from other trials. We need better and more predictive biomarkers and improve bioanalytical techniques which are adapted to handle small sample volumes and alternative matrices other than blood. And finally, better tools and methods for applying biosimulation will continue to enhance our ability to develop drugs more effectively in this population. We know that there are numerous reasons for pharma not to aggressively pursue development in pediatrics. In most circumstances, pediatric indications are not going to be particularly profitable and actually may represent a distraction to the development team who is very focused on gaining approval in adults, which is hard enough to do. So overall, we know that pediatrics in the U.S. market represents only about 7% of sales, and beyond this, there are ethical and legal risks and important challenges in the operational execution of studies. To address the market failure, regulatory agencies globally have introduced a number of important policies, and these have included policies which serve as both incentives, such as the 6 months market exclusivity available in the U.S. for studying pediatrics, as well as penalties or other requirements to address pediatrics in the approval process. So this slide summarizes the positive impact that these regulations have had over time, going from the early 70s of only about 22% of drugs having pediatric information in the label, which has increased to about half of these drugs in 2009. And we've seen similar trends in Europe. So it would appear that these regulations have had a positive impact. However, it should be noted, and numerous authors have identified neonates as an important and remaining underserved population. So we know that many pediatric programs exclude neonates, they're granted waivers from regulatory agencies, and otherwise fail to study this population in clinical trials. Due to the difficulties of pediatric development, the concept of extrapolation or bridging from adults was proposed a number of years ago. This slide illustrates an early representation of these concepts and really helps to guide drug developers in formulating a development pathway that is scientifically robust, yet operationally feasible. In cases where disease progression and pharmacology supports a similar response to the drug, it's not always necessary to conduct efficacy trials in children. Instead, it's possible to use robust PKPD approaches when one has the luxury of a strong pharmacodynamic marker to avoid efficacy studies, or if an exposure-response relationship has been established in adults and it's reasonable to apply this to children, to simply dose in pediatrics in order to achieve similar plasma exposures. So this approach provides a framework for making pediatric programs more efficient where possible while retaining scientific rigor. And from this framework, methods to bridge and scale from adults to children has become a significant emphasis and has led to significant advances in both tools and in methods. So this figure will be well known to this audience and illustrates the numerous physiological changes that occur in children which can have an impact on drug disposition. So it's well known that maturation of metabolic capacity and renal function can have a big impact. In addition, changes in body composition can alter volumes of distribution, and changes in GI function can also impact processes like absorption. So each of these factors, depending on the specific drug, needs to be accounted for in bridging from adults to kids. And fortunately, because this information has become better characterized over time, approaches to incorporate these factors using pharmacometric methods, for example, have become rather commonplace. Now, this figure only describes the impact of age on PK, but we all know that there are pharmacodynamic considerations as well. These may include factors such as the number of drug-related receptors present as a function of age, or things like the quality or function of those receptors compared to adults. and oftentimes these factors may be unknown in advance. So based on its very nature, quantitative pharmacology is uniquely positioned to play a major role in pediatric development. Because pediatric development is an area where data are sparse and limited, methods which can fill in or fill the void of empty spaces in the data are needed. So quantitative pharmacology is designed to do just this, and certainly has proven to be one of the most important tools available in the armamentarium for facilitating pediatric development. But we know that pediatric development is still tough. The approach is never straightforward. It's always done on a case-by-case basis and really requires multidisciplinary expertise in sciences and in the execution of studies, which includes areas like pediatric clinical pharmacology, quantitative methods, and regulatory science. And why is this important? Well, it's often an opportunity that's not capitalized on for a number of reasons. For example, inherent challenges with development capacity within companies, capability or capital constraints. The result, however, is oftentimes lost opportunities and delays in the introduction of important new medicines to the pediatric population. One growing trend that may help expedite pediatric development includes options to partner pediatric components of programs to experts who are specialized in this area. This may include partnering of entire programs or of particular aspects of programs such as formulation development and manufacturing, clinical development, all the way through to sales and marketing. So with that, I'll turn it over to Craig to talk about oseltamivir development in infants for influenza.
Thanks, Patrick. So I guess I'll now describe the case of the development and approval of Tamiflu for infants under 1 year of age. So why talk about this case? Firstly, I do believe it showcases a number of translational and quantitative pharmacology strategies and lessons learned that have broad applicability in pediatric development. Secondly, the approach described actually did result in successful and harmonised approval for oseltamivir dosing in babies in Europe and also in the US. Thirdly, the case is well referenced and will enable further reading for those interested. And finally, whilst you may think that the extensive on-market experience in older children and adults with Tamiflu would make this a simple case, the story is much more complicated, as you will hear. Tamiflu is the brand for oseltamivir, an oral neuraminidase inhibitor now indicated for the treatment of acute, uncomplicated influenza in patients 2 weeks of age and older. Of important historical context, the treatment indication for Tamiflu was initially restricted to children greater than 1 year of age due to a toxicology signal attributed to its prodrug, which you'll hear about a little later, in juvenile rats. However, the development for infants was reignited. It was fueled by a concern relating to both avian influenza and pandemic influenza, which highlighted the fact that there were no available influenza treatments for babies. In addition, emerging off-label use for babies started to demonstrate the potential safety of oseltamivir in this patient group. Even so, it's true to say that there was especially heightened diligence for moving into infants in light of the juvenile toxicology findings. Now for some pharmacology. Tamiflu is oseltamivir, an ester prodrug that's converted to its active metabolite, oseltamivir carboxylate, within the liver. During influenza infection, virus infects epithelial cells via rate beta. Infected cells produce new virus, adding to the virus pool according to the rate V. It goes into effect more epithelial cells. epithelial cells die and the immune system clears virus. And oseltamivir carboxylate reduces T on this diagram, or the rate of virus production. Reduction in virus production has benefits in terms of cytokine profiles, which can be seen in more detailed published mechanism-based models. But ultimately, treatment with oseltamivir results in reduced viral shedding, resolution of signs and symptoms of influenza infection, and potential reductions in secondary complications. In terms of relevance to pediatric development, available phase 3 studies with oseltamivir in adults and children greater than 1 year of age, it's very important to consider and reasonable to assume that children, when compared to adults, have similar disease progression and response to intervention. To some ADME properties. So when oseltamivir is administered, it's converted rapidly and extensively in the liver to oseltamivir carboxylate via human carboxyl esterase 1 or CES1. CES1 metabolizes ester substrates to organic anions. They're often substrates for UGT and organic anion transporters like MRP and BCRP. Oseltamivir carboxylate is a little bit of an exception, and as a consequence, it's trapped within the hepatocyte and slowly leaches out, resulting in flip-flop pharmacokinetics and a half-life that actually enables twice-daily dosing. Both the prodrug and metabolite are not highly protein-bound and are distributed in total body water. They also circulate in plasma and are eliminated via glomerular filtration or tubular secretion. So in terms of relevance to infants under 1 year of age, it was these ADME characteristics that initially painted a picture of a drug that could be quite sensitive to ontogeny or developmental changes in PK. Since it's known that significant changes in total body water occur over the first year, there are rapid changes in liver to total mass, body mass ratios over that first year, as does, as Patrick alluded to earlier, maturation of renal function, especially relating to tubular secretion. And at the time, it was quite new for us to consider that there was data that was suggesting that carboxyl esterase itself may be developmentally regulated. That is, a surge of activity or capacity during the first month of life. It was this competence of carboxyl esterase 1, which is of great interest, has actually raised questions as to the anticipated safety margins of oseltamivir, or the prodrug, which was the moiety of greatest interest in terms of safety, and also whether adequate metabolite concentrations could be produced, which also raised efficacy and resistance concerns or potential concerns for the very young. But before I highlight further the evolution of the under 1 program, I'd like to outline a couple of key messages I'd like you to be tuned into. Firstly, it's essential to develop a pediatric clinical pharmacology strategy. A pediatric clinical pharmacology strategy is a cornerstone piece within any overarching pediatric program. It must emphasize integration and deliberately encourage communication and linkage with commercial, regulatory, clinical, pharmacovigilance, and drug product strategies. A pediatric clinical pharmacology strategy should be enabling of clinical trial design and stewardship, strategy to define and execute quantitative analyses to enable dose justification, and should encourage integrated thinking across a program to provide supportive pharmacological and quantitative evidence that it can assist in providing meaning and certainty from adjacent disciplines such as non-clinical, pharmacogenomics, safety, even health economics, that ultimately can benefit pediatric development. The formal components that constituted the development program for oseltamivir in infants really began with the initiation of study CASG-114, or study 114 I will describe it as. It was a study that was led by the National Institutes of Health with input from Roche and Genentech using Roche's proprietary pediatric formulation. Subsequently, and at the encouragement of the European regulators, an additional study was performed by Roche known as Study WP22849 or Study 849 that added to the available patient database. And due to concerns over dosing accuracy of the initial proprietary formulation, explored the application of an extemporaneous formulation. This was followed by filing and registration of an actual novel proprietary formulation of oseltamivir in infants in the US and then in the EU. Commensurate with the high level of uncertainty at the time and considering the juvenile toxicology findings, Study 114 was designed with significant controls and monitoring, as would be considered for a new molecular entity with much less data in older children and adults. The phase 1/2 study was adaptive in design, evaluating PK, safety, virology, incorporated key clinical pharmacology features, including adaptive age de-escalation with DSMB oversight, which incorporated within age cohort dose adaptation based on both safety and PK. In cases where minimum numbers of subjects failed to meet pre-specified AUC targets, decision criteria for increasing, decreasing the dose, or recruiting more subjects at the same dose were implemented. The PK sampling strategy was also kept minimal via consideration of optimal sampling. Ultimately, these innovative clinical pharmacology contributions were key to the successful design and execution of Study 114. As briefly mentioned, Study 114 involved the use of the Roche proprietary pediatric formulation at the time. It was this formulation that was approved in children greater than 1 year of age, but given regulatory concerns relating to the dosing accuracy of this formulation for the very low volumes in the youngest infants, an additional study was conducted, and again, that's 849. So, like Study 114, Study 849 was also a phase 1/2 study evaluating PK, safety, virology in infants under 1 year of age. A POP-PK model derived from interim data from Study 114 was used to assist in dose recommendations for the new 849 trial. Assumptions on the new formulation performance, as well as remaining concerns on safety, resulted in lower doses being evaluated in the youngest infants in this study. Not only did Study 114 POP-PK model-derived simulations support dose selection, but they actually enabled a simplified, non-adaptive, parallel design that was substantially easier to execute than Study 114. And optimal sampling methods, again, supported limiting the PK sampling requirements for this study. The next part of the evolution of the under-ones program for Tamiflu involved the decision to file for the US, which was made despite Study 849 still ongoing. The urgency to file was largely fueled by unmet need. Coping with ongoing and incomplete trials due to operational challenges, the presence of differing health authority perspectives on dosing and data package requirements, and dealing with the reality of imperfect data are common challenges for most pediatric programs. For oseltamivir, there were particular challenges with the decision to file, including differing health authority initial perspectives on dose, data package and formulation requirements, as well as key challenges with how to deal with the limited data in hand across two studies where there were many similarities but also key differences. These facts underscore the difficulties in developing medicines in children and also the importance of creative quantitative pharmacology approaches to assist in navigating such potential roadblocks and challenges. Indeed, all of these features inform the global filing strategy, including its timing, the sequence, data packages, and scientific approach to thread the needle through all of these challenges. The next component of the pediatric clinical pharmacology strategy was indeed the scientific approach to establish dosing in babies in less than 1 year of age. The approach was predicated on three main pillars, requiring pooling of both the 114 and the 849 studies. The first step was to develop a POP-PK model to assist in plausibly addressing the heterogeneity in the studies that might impact on PK, such as the differing dose and formulation, and other developmental covariates. The PK model was then able to be used later as an engine to assist with simulation of both the prodrug and metabolite exposures for infants under the age of 1. The second step was to provide due diligence around exposure response in infants. If exposure response was shown to be different than older children and adults, then dosing would be based on infant-specific PKPD and PK safety relationships. If exposure response could not be demonstrated to be different in infants versus older children and adults, then this was considered to be grounds for it being reasonable to perform PK bridging to older children and adults to support dosing. The final step was taking the information derived in step 2 and using the POP-PK model to simulate candidate regimens that, in conjunction with the perspectives from other team functions, would be used to assist in recommendations for doses in babies. The penultimate analyses of the full 114 and 849 studies and the entire strategy that I've just outlined that eventually led to the approval has been published, and I direct you here for further detail. But to hit some of the highlights in terms of the PK, a 3-compartment population PK model was developed and accurately described the concentration time course of oseltamivir and oseltamivir carboxylate. Whilst allometric scaling used body weight adequately to describe the pharmacokinetics of the prodrug alone, and the metabolite apparent clearance and volume also linearly increased with age across that first year. Despite exhaustive covariate exploration, no relevant impact of gender, ethnicity, race, or impact of post-conceptual age could be detected. And ultimately, the PK model findings were plausible and also consistent with the literature. And importantly, no study differences in bioavailability could be detected. As far as the PKPD and PK safety evaluations across pooled studies, there's no new information that was able to be gleaned that would suggest a different exposure-response relationship in infants versus older children or adults. Consequently, the POP-PK model was used to examine candidate dosing regimens for infants under the age of 1 year and the ability of such regimens to achieve similar metabolite AUC, Cmin, and Cmax values and provide confidence that the prodrug exposures were not of a safety concern. By way of example, here are the oseltamivir carboxylate AUC simulations for 3 milligrams per kilogram twice daily for infants less than 1 year of age, to the left of the dotted line. To the right are the approved doses in children and adults, up to 75 milligrams twice a day. And a dose of 150 milligrams twice a day is also shown to be safe and effective and well tolerated in phase 3 studies in adults. Doses of 225 and 450 milligrams twice daily that were safe and well tolerated in a Q2 study are also shown in this schematic. A dose of 3 milligram per kilogram twice daily was considered necessary to ensure adequate metabolite exposures across all infants less than 1 year of age, especially for those infants less than 1 month of age where the PK variability was highest. The PK variability was considered to be a function of ontogeny, reflecting variability in maturation of the features that we've spoken about: carboxylesterase surge, renal function and immature tubular secretion and changes over that first month, and rapid development in body composition, including liver and total body water. Not shown here was that across all infants, the prodrug exposures were well within the clinical safety margins and acceptable. And it was this pediatric clinical pharmacology package, augmented with supportive information from other disciplines, that was the thrust of the supplemental NDA for FDA that was accepted. So, moving on to the European filing, which occurred after the US filing. The general data package and argumentation was very similar to what I've just outlined, but it didn't include any interim data. But in addition, it also included some further quantitative pharmacology. One important addition was the incorporation of additional toxicology information. Specifically, an adult and juvenile marmoset study was conducted and confirmed that carboxylesterase activity was developmentally regulated. A PBPK model was developed to assist in bridging the data from both adults and juvenile marmosets in an iterative fashion with human adults and infants. The PBPK model was shown to adequately simulate both the prodrug and metabolite in both species, as well as capture the age dependencies. And the EMA considered the additional PBPK model as important, as it supported the biological meaning behind the more empiric observations from the POP-PK model. EMA drew reassurance from the PBPK model in that it provided supportive evidence that human infants should have sufficient prodrug turnover to have adequate phosphatidylate carboxylate for efficacy and also acceptable safety margins for both the prodrug and the metabolite. Ultimately, I believe this showcases a number of translational and quantitative pharmacology strategies that have broad applicability in pediatric development. The case highlights that pediatric clinical pharmacology strategy enabled clinical trial design and stewardship, strategy to define and execute quantitative analyses to enable dose justification, and provided added value, such as enhancing biological meaning, that resulted in successful and global approval for the first medicine to treat influenza in patients 2 weeks of age and older. So at this juncture, I'd like to now hand back over to Patrick, who will continue the discussion.
Great, thanks, Craig. So a common question is, when should pediatric trials be started? And when they're conducted really depends on the nature of the drug and the indication. We know that 90% of drugs that enter development won't eventually make it to market. So it's a common view to wait until safety and efficacy has been established in adults prior to exposing children to these new medicines. However, guidance on when trials can or should be initiated is provided by ICH E11 and summarized in this table. According to the guidance, appropriate timing to initiate studies depends on the indication and the state of existing therapies. So, for example, for indications affecting children exclusively or predominantly, the entire development program can be conducted in the pediatric population, starting with phase 1 or phase 2 studies, oftentimes beginning with a phase 1 healthy adult volunteer study, if that's relevant. For serious diseases that affect both children and adults for which there are no adequate treatment options, the recommendation is for studies in children to be conducted early, after some initial safety and tolerability data have been attained in the adult population, as well as some reasonable evidence of potential benefit. In other areas, when indications are present in both adults and children, which may be less serious diseases or where adequate treatment options exist, it's recommended for pediatric trials to be initiated later, after more substantial experience is gained in adults. What's important to recognize then is that the ability to bridge from adults to children, and the amount of available data and the assumption of risk changes as pediatric programs are initiated earlier. Yes, there's more risk, but these are drugs that have the highest probability to serve an unmet medical need and from which will benefit the most from improvements in science and innovation. So Craig shared an interesting case of pediatric development in influenza. And while that is a mature and well-developed case, oseltamivir was approved first in adults and was on the market for a number of years prior to gaining approval in the infant population. A very different and emerging area of activity for pediatric development is in the treatment of RSV infection. There are a number of compounds now in clinical development for this indication, and based on the ICH criteria, which I showed on the last slide, RSV compounds would clearly fall into a category where very early study in pediatrics would be appropriate. RSV is a respiratory viral infection, primarily an issue in children who are 2 years of age and younger. All children will be infected, but a subset will go on to develop significant disease that may result in hospitalization or mortality. It's also been associated with the development of recurrent wheezing and asthma in children later in life. There are limited current treatment options, and treatment is generally supportive in nature. So clearly a high unmet need area in infants, and also presents an interesting opportunity and model to apply some of the same translational techniques which Craig spoke about and which have been developed and improved over the past decade. Some of the options for developing RSV compounds, or from a clinical development plan perspective, are summarized on this slide. In most cases, one would enter humans in a traditional phase 1 adult SAD-MAD study. Following the study, there are then several options. One could go then into an elderly or immunocompromised population to demonstrate proof of concept, or to utilize a recently developed RSV human challenge model where healthy volunteers are inoculated with an attenuated viral strain, allowing for dose-ranging PKPD studies to be conducted and to demonstrate proof of concept. So the question then from there is when to start pediatric studies, knowing in particular that the population of relevance is infants who are less than 2 years of age. So Alios is a sponsor we've had the opportunity to work with in the early development of ALS 8176, which is a novel pan-genotypic nucleoside analog inhibitor of viral replication. We also appreciate that they've given us permission to share this example despite the fact that the drug is still being developed and is in the early stages. ALS 8176 is administered orally as a prodrug, is activated intracellularly to the triphosphate form, which results in inhibition of viral replication via chain termination. Now, the approach that was taken in this case was to conduct a healthy volunteer SAD-MAD study and then in parallel begin collecting PK data in infants. PK modeling with allometric scaling was used to help justify dosing in order to achieve concentrations that were expected to be in the therapeutic range based on preclinical animal studies. And this work was conducted, incidentally, in collaboration with Certara. So in essence, after dosing in approximately 60 healthy volunteers, The next patient who received this drug was an infant. And to our knowledge, this is one of the first examples whereby a small molecule has successfully taken this approach to go into infants so early in development. Perhaps it has been done previously, maybe in other therapeutic areas, and we're not aware. In parallel, as I mentioned, the adult healthy volunteer challenge model was conducted with the goal of generating exposure response data, which would be useful to show proof of concept and further define exposure response relationships. These are the top-line results of the challenge study, which were published in the New England Journal of Medicine. While not an ideal exposure response data set, particularly due to the expense of this particular type of study, the results demonstrate potent antiviral activity and enabled us to construct exposure-antiviral relationships. Because this is an antiviral, an assumption is that's not unreasonable was to assume antiviral activity would be similar in adults and pediatric patients. The results of these data sets allowed the construction of a PKPD viral kinetic model for RSV in adults, which is summarized on this slide. Not shown here is the pharmacokinetic model describing plasma concentrations systemically, but the model links plasma concentrations to lung triphosphate concentrations, which is the entity that acts to inhibit viral replication. And from this, we're able to craft an exposure response relationship. To provide initial estimates of PK and the impact of dose on inhibition of viral replication, standard allometric scaling of clearances and volumes, including incorporation of a renal maturation factor, was used. And this was utilized to estimate a phase 1 starting dose in pediatrics that had sufficient safety margins to preclinical species and to exposures obtained in adults, but was also expected to be in the low therapeutic range. Because this model initially was 100% based on adult data without any pediatric information, it was updated and adjusted over the course of the study as data were collected. So overall, this example demonstrates the application of many of the same tools which Craig described for oseltamivir, however applied in a way to support going into infants very early on in a program. Without a solid clinical, preclinical, and modeling and simulation approach, this likely would not have been possible. So to summarize, there are many challenges associated with bringing drugs into the pediatric population early in the development process. Obviously, the earlier this is done, the lesser information there will be available for guidance. The ability to extrapolate will vary. However, the opportunity, if done carefully and thoughtfully, is the potential to address important pediatric diseases sooner. This includes diseases where no acceptable treatment options exist, including orphan diseases, diseases in the oncology arena, and even in medical countermeasures, where the current approach is approval based on the animal rule, where clinical trials are not possible. We know that there are constraints on capacity, expertise, and capital, which also may delay the availability of pediatric therapies, and these can potentially be addressed via partnering, outsourcing, and creative licensing approaches to make it happen. And finally, newer tools and approaches are clearly needed. There have been important advances, but more are emerging. These include some interesting approaches being explored, such as approaches for microdosing in pediatrics to characterize pharmacokinetics, further development of quantitative pharmacology methods, which include top-down and bottom-up approaches, as well as advances in study design. These innovations need to continue, and fortunately, there are many opportunities to innovate, which can happen in the pre-competitive space, whereby collaboration and sharing of information and discussions such as this webinar can all help to promote and facilitate bringing new medicines to children more quickly. So with that, we'd like to thank you for your attention and would be happy to entertain any questions or any comments from the audience. Thank you.
Okay, thanks so much, Patrick and Craig. As Patrick mentioned, we'd love for our audience to submit their questions to us through the Q&A panel. Looks like we've got our first question from our audience. You had mentioned that there's a growing trend of outsourcing pediatric development as well as the opportunity for pre-competitive collaboration. What possible models have you seen working in this area?
So maybe I'll start and Craig can pitch in. So there have been a number of announcements of partnerships which have related to manufacturing arrangements, formulation development. But I think one of the more interesting aspects that has been discussed is actually the partnering of the pediatric indications out to a specialty group, and essentially the entire pediatric development program can then be initiated and run separately with through a partnering relationship. And I think that's a pretty exciting potential approach for this to work. We've seen smaller examples of taking bits and pieces of programs, saying outsourcing the PIP, for example, which is difficult for internal development teams to deal with at times, and bringing in experts or having arrangements where this is done by third parties. So, I don't know, Craig, anything to add to that?
I'll probably just add about the pre-competitive space again, and just to underscore that. I think clearly the pediatric development community globally is extremely interested at advancing new methods, and I think an example is the PAMPers program in Europe, where microdosing and the application of AMS has been applied. I think there's some good publications which have come out from various consortia in those areas. So I think that there's great opportunity, again, as I think you stated, Patrick, in environments such as this with people on this call to come together and actually look for ways to continue to collaborate and to come up with new approaches. I think that there is a great opportunity in the quantitative pharmacology space to continue to move that along. So that would probably be the only additional thing that I'd add.
Okay, someone from our audience would like to know, you mentioned microdosing as a potential tool to be used to determine PK in children. The method works with radioactivity, and although the doses are extremely low, I'd expect resistance in using marked microdoses in a pediatric population. Do you have any experience that you could cite in this issue?
So that's a great question, and in fact there are two publications that I'm aware of, and the studies were done in Europe where this has been successfully done, and both involve the use of acetaminophen. So one of the publications was by Colin Gardner, and in both cases, right, they were successfully navigated through the approval of the ethics committee as well as through regulatory agencies. So both of those, and I think that in our additional reading there may be references to those publications, which have both come out in the last couple of years.
Someone else would like to know, when building pharmacokinetic models, are juvenile toxicology data or adult human PK data better predictors of pediatric PK? So
it's a really good question. I'll take a first stab at it, and Patrick will let you move on from there. So I think that there's no hard and fast rule. I see both as absolutely complementary. If you could imagine looking at trying to land on an aircraft carrier, they're effectively the bottom-up and top-down approaches to be applied. So I think that the concept of, if we take the top-down approach, the concept of trying to hardwire allometric principles even into POP-PK models early in a development program, and really leverage whatever data we can to try and extrapolate out of adults is clearly very, very helpful. And then to also augment that with from the bottom-up approach from juvenile models and non-clinical models, and asking how the two results actually reconcile. In the situation of the case which I presented for Tamiflu, where there's some good references, again for further reading, the PBPK model that was developed did a really honest job, but I would consider in that situation it was really the adult data coming down that probably had the greatest deal of strength, but it was the PBPK that was really the richness in providing the meaning behind it. So in that situation, it was probably playing more to the top-down approach versus the bottom-up approach. But again, both of them were very complementary. Patrick, I don't know if you'd like to add.
I think it's as always a case-by-case basis, right? It depends on the compound, how it's handled, and it also depends on the preclinical species that you're dealing with.
Someone would like to know, someone asks, it's been my experience that by having pediatrics flagged as a key patient population when you go into humans, you can start to future-proof your development program. What are some of the efficiencies that you see can be gained by this approach?
Patrick, did you want to, I'll add a couple. I think as I mentioned on the prior question, you know, I think just innately thinking about pediatric development really early, even with your early studies in adults, you start to think about how your POP-PK model might actually be developed. And in that regard, you are consciously looking at opportunities to enable that population later on in development. So you're thinking about optimal sampling. You're thinking about, again, hardwiring allometric principles into an existing POP-PK model, because whilst it may not be critically important right at the moment, you are saving time potentially later on as you're looking at trying to extrapolate out of into younger age groups. I guess the other piece is thinking very early on about your bioanalytics even. The sensitivity of the assays which are being used, being able to cope with some of the other operational challenges which you get in pediatrics. So not only optimal sampling in terms of numbers of PK samples, but also the blood volumes and plasma that are required for supporting PK. In the same vein, also thinking about PD. So what are the kinds of covariates that one would be needing to think about from a disease progression perspective, and how they might be portable later on during the development program. Patrick, I don't know if there's anything else that you want to add.
I guess the only thing that comes to mind is that really thinking about pediatrics early is no different than thinking about any other special population early, right? And beginning really with the end in mind and knowing what's around the corner, and start designing and collecting the data needed to enable use in those populations. So I would just say pediatrics is certainly on the list, but it's not the only one that's on the list.
Okay, it looks like we have one final question from the audience. This is speaking to the oseltamivir case study. Why was the juvenile rat finding based, was the juvenile rat finding based on actual toxicological studies in juvenile animals or based on reproductive studies? And why did you choose to use a marmoset as your animal model?
Yeah, excellent questions. So yes, it was based on real juvenile toxicology findings. Is the answer to the first part of that question. And why the marmoset? So the marmoset was considered to be the most appropriate model when it came to carboxylesterase maturation. So that was considered to be the most appropriate model for getting down into infants under the age of 1. So and particularly the lowest age ranges.
Looks like we have one final question. It's great to see so many questions from our audience. When developmental maturation functions for estimating the first doses, was a generic maturation function, aka sigmoidal, used from the literature since no data was available for this age group? Are the generic functions better for renal or hepatic function?
So for our example, all we had to go on was basically we knew renal clearance of the drug from adults, and then we applied that percentage of renal clearance using a generic maturation function that's published in the literature. So I'm not sure what else one could really do other than that at that stage in development. And then the second question as far as maturation functions for renal or for hepatic, I think it depends on the compound. My impression, certainly not an expert in this area, but my impression has been that it's a little easier to predict renal function, and that seems to be more well established and developed. There are just so many other things that can go in, like transporters and CYP metabolism and so on, that can impact hepatic scaling. That at least that's been my impression. But would certainly invite anyone from the audience to chime in who knows this in more detail.
Finally, someone would like to know whether the PBPK model was requested by the EMA or whether it was volunteered.
Yeah, a good question. I can't answer that. I don't know the detail, the very specific detail there. So yeah, I'm unable to answer that.